.gov, and his phone number is 301-796-6248. My name is Alberto Papo, and I will be chairing today's virtual meeting. I will now call the morning session of the Pediatric Oncology Subcommittee of the Oncologic Drugs Advisory Committee to order. We'll start by going down the meeting roster and introducing ourselves. We will once again use a call-respond method in which I will call the panel's member to prompt the member to speak, and the panel member will have a chance to introduce him or herself into the record. So we will just wait for the slides to load to show the pictures of the panel members. David Mitchell?
Yes, doctor, thank you. I'm David Mitchell. I'm a consumer representative. I'm also a cancer patient with multiple myeloma.
Thank you. My name is Alberto Papo. I'm a pediatric oncologist at St. Jude Children's Research Hospital, and I'm the chairperson of the Pediatric ODAC. Dr. Cheng?
Good morning, Jonathan Cheng. I'm the industry rep, and I'm with Merck Pharmaceuticals.
Dr. Catherine Bollard?
Yes, hi, I'm Catherine Bollard. I'm from Children's National and the George Washington University in Washington, D.C.
Dr. Stephen DuBois?
Hi, I'm Steve DuBois from Dana-Farber Boston Children's, a pediatric oncologist.
Dr. Ira Dunkel?
Good morning. My name is Ira Dunkel. I'm a pediatric neuro-oncologist at Memorial Sloan Kettering Cancer Center in New York.
Dr. Julia Glade-Bender?
Good morning. I'm Julia Glade-Bender, also of Memorial Sloan Kettering in New York, and I am a pediatric oncologist.
Dr. Richard Gorelick?
Good morning, everyone. I'm Richard Gorelick, the division head of pediatrics at MD Anderson Cancer Center in Houston, Texas.
Dr. Theodore Leitch?
Good morning. I'm Ted Leitch, a pediatric oncologist at UT Southwestern Medical Center in Dallas, Texas.
Donna Ludwinski?
Hi, Donna Ludwinski from Solid Kids Cancer in New York. I'm a patient representative.
Dr. Andy Kolb?
Yeah, hi, this is Andy Kolb. I'm a pediatric oncologist at the Nemours Center for Cancer and Blood Disorders in Wilmington, Delaware.
Dr. Catherine Janeway?
Hi, this is Katie Janeway. I'm a pediatric oncologist and sarcoma specialist at Dana-Farber and Boston Children's in Boston, Massachusetts.
Dr. Nainesh Kamani?
Hi, good morning. I'm Nainesh Kamani, pediatric immunologist and bone marrow transplanter at Children's National in Washington, D.C.
Dr. Toby McDonald?
Good morning. This is Toby McDonald. I'm a pediatric neuro-oncologist at Emory University and Children's Healthcare of Atlanta.
Dr. Leo Mascarenas?
Good morning. I'm Leo Mascarenas. I'm a pediatric oncologist at Children's Hospital Los Angeles and the University of Southern California.
Dr. Will Parsons?
Hi, I'm Will Parsons. I'm a pediatric oncologist and the Deputy Director at Texas Children's Cancer and Hematology Centers and Baylor College of Medicine in Houston, Texas.
Dr. Elizabeth Rates? Elizabeth, are you on mute?
No, sorry. Good morning. This is Elizabeth Rates. I'm a pediatric oncologist at New York University.
Dr. Nita Seibel?
Hi, I'm Nita Seibel. I'm a pediatric oncologist. I'm in the Clinical Investigations Branch of CTEP at the National Cancer Institute.
Dr. Malcolm Smith?
Good morning. I'm Malcolm Smith in the Cancer Therapy Evaluation Program at the National Cancer Institute, a pediatric oncologist. Thank you.
Dr. Latoya Bonner?
Good morning. This is Latoya Bonner. I am the DFO for this meeting.
Dr. Gregory Riemann?
Good morning. I'm Gregory Riemann, Associate Director for Pediatric Oncology and the Oncology Center of Excellence and CEDAR's Office of Oncologic Diseases.
Dr. Denise Casey.
Good morning, everyone. I'm a pediatric oncologist at FDA, Division of Oncology 3.
Dr. Leslie Doris.
Hi, I'm Leslie Doris, FDA Division of Oncology 3 Pediatric Oncologist.
And Dr. Megan Zimmerman.
Good morning, this is Megan Zimmerman. I'm a pediatric oncologist and clinical reviewer at FDA.
Thank you very much. For topics such as those being discussed at today's meeting, there are often a variety of opinions, some of which are quietly, strongly held. Our goal is that today's meeting will be a fair and open forum for discussion for these issues and that individuals can express their views without interruption. Thus, a gentle reminder, individuals will be allowed to speak into the record only if recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the Government in the Sunshine Act, we ask that the advisory committee members take care that their conversations about the topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with the FDA about these proceedings. However, the FDA will refrain from discussing the details of this meeting with the media until its conclusion. Also, the committee is reminded to please refrain from discussing the meeting topic during breaks or lunch. Thank you. We will now proceed with the FDA introductory remarks from Dr. Greg Riemann.
Good morning. I'd like to welcome the expert advisors as well as our pharmaceutical company sponsors to day 2 of the Pediatric Subcommittee of the Oncologic Disease Advisory Committee. Again, as in the past, our focus for these meetings are really to accelerate the timely development of novel anti-cancer agents with potential applicability to one or more pediatric cancers. At the present time, the only pediatric legislative initiative that is relevant to cancer drug development in children is the Best Pharmaceuticals for Children Act. We will hear presentations and discuss 2 products in early development under INDs in an attempt to maximize the agency's authority under the Best Pharmaceuticals Best Pharmaceuticals for Children Act, which is a voluntary program utilizing the written request mechanism. The products for discussion will be a novel engineered cell therapy from CD30 CAR T-cell product from Tessa Pharmaceuticals and a menin inhibitor SNDX5613 from Syndax. Company presentations and expert panel discussions and recommendations will serve to help inform the review divisions of the Office of Oncologic Diseases and the Office of Tissue and Advanced Therapies in CBER, as well as the Oncology Center of Excellence, as to whether written requests for pediatric assessment should be issued based on the degree of unmet clinical need and potential public health benefit to children, the quantity and quality of both non-clinical and adult clinical data to support pediatric investigations, and finally an assessment as to whether or not appropriately designed clinical trial in children provides a favorable benefit risk. So again, I would like to thank you all and acknowledge your patience with the technology that we are forced to work with during. And again, we appreciate your participation in this meeting. Thank you.
Thank you very much, Dr. Riemann. Dr. Latoya Bonner will now read the conflict of interest statement for the meeting.
Thank you, sir. The Food and Drug Administration is convening today's meeting of the Pediatric Oncology Subcommittee of the Oncologic Drug Advisory Committee under the authority of the Federal Advisory Committee Act, FACA, of 1972. With the exception of the industry representatives, all members of the committee and temporary voting members of the subcommittee are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of the subcommittee's compliance with the federal ethics and conflict of interest laws covered by but not limited to those found at 18 U.S.C. Section 208 is being provided to participants in today's meeting and to the public. FDA has determined that members of the committee and temporary voting members of the subcommittee are in compliance with federal ethics and conflict of interest laws under 18 U.S.C. Section 208. Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflicts when it is determined that the agency's need for special government employee services outweighs his or her potential financial conflict of interest, and when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Related to the discussions of today's meeting, members of the committee and temporary voting members of the subcommittee has been screened for potential financial conflicts of interest of their own as well as those imputed to them, including those of their spouses or minor children, and for purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contract grants, quotas, teaching, speaking, writing, patents and royalties, and primary employment. For today's agenda, information will be presented regarding pediatric development plans for 2 products that are in development for an oncology indication. The subcommittee will consider and discuss issues relating to the development of each product for pediatric use and provide guidance to facilitate the formulation of written requests for pediatric studies, if appropriate. The product under consideration for this session is CD30.CAR-T, presentation by Tessa Therapeutics. This is a particular matters meeting during which specific matters related to CD30 CAR-T will be discussed. Based on the agenda for today's meeting and all financial interests reported by the committee members and temporary voting members, conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208, B3 for Drs. Ira Dunkel, Theodore Leitch, and Leo Mascarenhas. Dr. Dunkel's waivers involve consulting interests with 3 companies for which he received remuneration between $0 to $5,000 per year from 2 companies and between $10,001 and $25,000 per year from a 3rd company. Dr. Leitch's waivers involves his employer's research contract funded by the Children's Oncology Group. Dr. Mascarenhas' waiver involves his employer's research contract funded by AstraZeneca. The waivers allow these individuals to participate fully in today's deliberations. FDA's reasons for issuing the waivers are described in the waivers document, which is posted on FDA's website at www.fda.gov/advisory-committees/committees meeting materials/drugs/default.htm. Copies of the waivers may also be obtained by submitting a written request to the agency's Freedom of Information Division, 5630 Fishers Lane, Room 1035, Rockville, Maryland, 20857, or requests may be sent via fax to 301-827-9267. To ensure transparency, we encourage all standing committee members and temporary voting members to discuss any public statements that they have made concerning the product at issue. With respect to FDA's invited industry representative, we would like to disclose that Dr. Jonathan Chang is participating in this meeting as a non-voting industry representative, acting on behalf of regulated industry. Dr. Chang's role at this meeting is to represent industry in general and not any particular company. Dr. Chang is employed by Merck & Company. We would like to remind members and temporary voting members that if the discussions involve any other products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all other participants to advise the subcommittee of any financial relationships that they may have with the firm at issue. Thank you.
Thank you very much, Dr. Bonner. Both the FDA and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the advisory committee meetings, the FDA believes that it is important to understand the context of an individual's presentation. For this reason, the FDA encourages all participants, including the applicant's non-employee presenters, to advise the committee of any financial relationships that they may have with the firm at issue, such as consulting fees, travel expenses, honoraria, and interest in the applicant, including equity interests and those based upon the outcome of the meeting. Likewise, the FDA encourages you at the beginning of your presentation to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your presentation, it will not preclude you from speaking. We will now proceed with Tessa Therapeutics' presentation.
Good morning. Thank you very much, Dr. Papa, Dr. Raymond, and members of the ODAC and member of the FDA. I'm Ivan Horak with Tessa Therapeutics in Singapore, and on behalf of the company, we are very grateful for the opportunity to present our CD30 CAR-T program here and get the input from ODAC members. So Tessa Therapeutics is a biotech company which is global but is headquartered in Singapore, which is focusing on the CAR-T technology and virus-specific T-cell platform. We received ARMA designation for relapsed and refractory classical Hodgkin lymphoma early this year, and we'd like to discuss this program at this meeting. So Hodgkin lymphoma is a highly curable disease, and the majority of the patients are cured through a first-line therapy. There are various variations of the first-line therapy, which is usually the combination of the multiple agents. And after a small fraction of the patients who relapse, they advance to high-dose chemotherapy with a bone marrow transplant. But the third and fourth-line therapy are usually reserved for anti-PD-1 antibody or brentuximab vedotin. But there is no approved treatment post-BV or PD-1 antibodies. Therefore, there is a high unmet medical needs for the small fraction of the patients who may benefit from novel therapeutics. So on this graph, it's a well-known phenomenon of the bimodal expression or incidence of the Hodgkin lymphoma, where the incidence is high in the young adolescent patients and then is increasing over the aging patient population. It is clear with a light bar that incidence, although is very high for young adolescents, but the death rate or mortality is significantly low, and then it's increased with the age of the patients. Now, in addition to Hodgkin lymphoma, which are universally CD30 positive, there are other subsets of the non-Hodgkin lymphoma which might be relevant for pediatric patient population, primarily anaplastic large cell lymphoma, which is almost 100% CD30 positive, and represent a meaningful fraction of the patients, pediatric patients with non-Hodgkin lymphoma. The second subgroup, which has an increased expression of the CD30, are the diffuse large B-cell lymphoma, which represent a meaningful subset of the non-Hodgkin lymphoma in pediatric patient population. Other CD30 positive non-Hodgkin lymphoma are probably less relevant for this discussion. On subsequent slide, you can see sort of a summary of the various aspects of the CD30 and lymphoma, so from a high-level perspective. So as I mentioned, CD30 is universally expressed on Hodgkin and Rhys-Sternberg cells. This is a very well-validated target, primarily through the antibody-drug conjugate BV, which got approval for Hodgkin lymphoma as well as for some subset of non-Hodgkin lymphoma. What is documented from BV and some other studies using cell therapy and antibodies, the CD30 is a safe target and actually is almost universally expressed on the various tumors, primarily of Hodgkin lymphoma and some non-Hodgkin lymphoma, but it can be expressed on activated T cells, small number of the eosinophils, and actually what is clear from the study which I will present on keratinocytes. T cell therapy, we believe, is an exciting opportunity to fill the vacuum for the patients or opportunity for patients who failed third-line therapy for non-Hodgkin lymphoma. So on slide number 9, you can see the picture design of the CD30 construct. So CD30 construct is a single-chain FV, which is was generated from HRS3 murine antibody. Has a linker which is CH3 linker, which has an antibody ADCC or FC receptor component, and then it's a transmembrane and cytoplasmic tail of CD28 and CD3 zeta chain. So it will be probably a classical type 2 CAR T cells. On the right side of the slide, it's a very general, high-level picture of the development of from PBMCs going through a CD3, CD28 activation, ultimately a transduction of CD30 and expansion of the T cells in the presence of the IL-7 and IL-15. So let me just tell you about the clinical experience, primarily in adults, but in a small number of the pediatric patients with this construct. So on the next slide, there are 3 clinical trials, one which was published in a totality, which is the first trial published in the Journal of Clinical Investigation in 2017 from Carlos Ramos. And this is interesting study because it's using CAR T for Hodgkin and non-Hodgkin lymphoma, but without lymphocyte depletion. And in spite of LD therapy, there are 3 out of the 9 patients achieved complete remission. The subsequent 2 studies, a study at the University of North Carolina and Baylor College of Medicine, are the sort of a main component of my presentation. So on this slide, it's the design and the number of the patients who were treated with this 2 parallel phase 1, phase 2 trial. So 59 patients were underwent procurement of the T cells. However, from these 59, 15 were not treated for various medical and non-medical reasons, which are very well articulated in the blue box. On the left side of this, you can see 44 patients who received infusion, 26 patients at the University of North Carolina, and 18 patients at the Baylor College of Medicine. So these are all patients with a classical Hodgkin lymphoma who failed multiple lines of prior therapies, and I will talk about it a little bit later. But what is important that there are 3 different types of the lymphocyte depletion. Small group of the patient at the UNC, they did not receive, they received only bendamycin as a lymphocyte depletion without fludarabine. And then additional 2 groups, one at the UNC, one at Baylor, they received combination with fludarabine. At UNC, combination of fludarabine with bendamycin, and in Baylor was a traditional flu-side regimen. So both 18 patients in both arms. So next slide, it's a demographic baseline of the patients who were treated by this protocol. So it is not surprised that the majority of the patients are at the initial diagnosis were stage 3 and 4. The median age of the patient is not really very surprising, slightly the predominance of male patients. What is surprising is the number of the prior therapies. Median is 8, but ranging from 5 to 17. 2/3 of the patients received bridging chemotherapy prior to receiving T-cell therapy. Majority of the patients received prior BV therapy, or 93%. Significant number of the patients received immunotherapy with anti-PD-1 antibody between 3/4 and up. Majority of the patients got high-dose chemotherapy and stem cell transplant and or support, and 25% of the patients received the allo transplant as well on the top of the prior therapies. The dose of the cell therapy was initial dose escalation started with 20 million cells going to 100 and 200 per meter square. At UNC, the start was a little bit high with 100 million cells per meter square going to 200. So on next slide is a high level of the clinical responses. So in the first column, you can see the total group, and then subsequent columns are showing according to a different lymphocyte depletion therapy. So going from the first high level, the response rate for patients who failed multiple lines of therapy was pretty high, but probably very similar to a response rate with the anti-PD-1 antibody and BV as a third, fourth line therapy. So 3/4 of the patients responded to the treatment, but what was surprising was the high level of the CI rate, 56% and 8% of the partial responses. So that really compares very nicely with the even less pretreated patients with BV or anti-PD-1. For instance, for illustration, anti-PD-1 antibody induced around 16% of 16 complete remission, and BV around 33%. What was surprising that bendamustine, a small number of the patients, had no response whatsoever. So really the fludarabine was important component of the lymphocyte depletion therapy. And looks like and seems in a small number of the patients that bendamustine with fludarabine it provide higher number of the complete remission compared to flu-cy regimen. So on this slide, I would like to show the swimmer plot showing the patients the duration of responses. On the left side, you can see, if it's visible on your screen, the previous therapies highlighting the sort of the key important prior therapies, including BV, anti-PD-1 antibody, and the transplant. And on the right side, you can see the duration of responses. And the median duration of response for patients with complete remission was pretty significant after first infusion with this follow-up, which is censored in February 2020, 9.5 months. 14 out of the 29 patients reached CR, they failed BV and PD-1 antibody, and 1 year survival of the group is 94%. So on the next slide, I would like to focus on the differences between the type of the lymphocyte depletion. So in the left upper panel, you can see progression-free survival of patients who received fludarabine containing lymphocyte depletion. So it means flu-cy or benda-fludarabine. On the right side is really the distribution between, on the top in a red line, patient who failed bendamustine fludarabine, and on the blue line, patient who received fludarabine cytoxan. And that significant differences in the progression-free survival. So 1 year PFS was 38% for total group. Now, if you compare the fludarabine bendamustine, is 57% of the patients have a PFS for 1 year, and flu-cy 21%. So it seems, again, small number of the patients, there are some curve separation. So the safety profile of the CD30 CAR was really impressively good compared to, let's say, experience the CD19 CAR therapy. And we can speculate what is the reason behind it, but let's just focus first on the toxicities. So majority of the toxicities are due to a lymphocyte depletion therapy, so driven by flu-cy or benda-fludarabine. There are a few things which really stood up in terms of the safety profile. So one is that cytokine release syndrome was reached only grade 1 and was all resolved spontaneously and didn't require any therapeutic intervention. The second one is very interesting, and this is 18 patients, or 41%, had grade 1 to grade 3 skin toxicity, which resolved spontaneously, were largely symptomatic, And on the biopsies, it looks like a spongiotic dermatitis with occasional involvement of the eosinophils. And very nicely documented by the study, showed that on a skin biopsy, there was by qPCR, low level of expression of the CD30 in the keratinocytes, may be responsible for this toxicity. No CNS toxicity was noticed. Now, one can speculate why the differences between the CD19 and CD30. One possibility is a low tumor burden because the CD30 is expressed only on Hodgkin and Reed-Steinberg cells, clearly a total tumor volume much lower than one would see with ALL or diffuse large B-cell lymphoma. Pediatric patients. So from the small group of the patient in phase 1, phase 2, 3 patients would account for pediatric category. 2 were 15 years old, 1 17 years old. They failed multiple prior therapies. 2 achieved a complete remission, 1 on bendamustine fludarabine and 1 on Flu/Cy. Unfortunately, 1 patient on Flu/Cy did not really respond to the treatment very well. And this patient was actually probably high risk anyway, required a bridging chemotherapy. 2 patients who got a complete remission, they waited for a cell therapy. These are bridging therapy. Again, the safety profile was not really different compared to adults. And again, primarily toxicity was driven by the chemotherapy. Interesting is the pharmacology and CD30. So CD30 and cell persistence was followed by the 2 methodologies, flow cytometry and qPCR by the copy number. So it is probably not surprising that the cell expansion and persistence was to some extent dose dependent. So you can see this high dose going up to 200 million cells on bendafludarabine. You can see significantly higher area under the curve. And there was pretty significant difference between the bendamustine and fludarabine containing regimen at the same cell level. Again, is a small number of the patients. And now, what is surprised that the persistence of the cells did not correlate with the responses. There's a clear dose response in the term of the cells. So the more cells you give, it's not surprising that the peak and the area than the curve will be higher. So it's a dose-dependent increase of the area than the curve. These are 3 patients, pediatric patients, where we have a very limited PK profile. So it's really hard to make too much from it. But it just showed that there's a pretty decent persistence of the cells over several weeks. So after this, I would like to go straight to the pivotal trial design, which we submitted to FDA for evaluation. And so this we call Chariot trial, which is for patients with relapsed and recurrent Hodgkin lymphoma. In this study, we expect to enroll 82 adults to reach 66 valuable patients. We expect 20% dropout, which is a little bit on the higher side. And we'd like to enroll at least 5 pediatric patients. It will be really interesting to discuss since we have a very limited experience with CD30 CAR. In the pediatric patient population, we would like to start and open for a treatment up to age 12. And if the safety and tolerability and efficacy justified and to go to a lower age population, although the incidence of and probability to enroll this patient population to a lower age, let's say up to 5, might not be very high due to high cure rate, as I mentioned. We expect to do a lymphocyte depletion therapy using bendamustine fludarabine. We plan to provide 200 million cells per square meter, and in pediatric patient population, we plan to dose based on a kilogram, primarily for a patient who are less than 60 kilogram weight. We open the opportunity to give a second dose, if needed, for the patients who have some level of response, so the lowest will be stable disease, but it can be done only between 3 to 6 months after the initial dose, and there must be a tumor biopsy to show that the patient has Hodgkin's lymphoma, which is CD30 positive. And then there will be long-term follow-up first on every 3 months basis, up to 24 months, and then every 6 months up to 15 years is required by FDA. So what is the primary endpoint? It will be response rate, which has to be assessed at 9 months follow-up. There is a multiple secondary endpoint, some clinical, some exploratory, looking at the performance of the CD30 cells, look at the immunogenicity, cytokine profile, immunological parameters, and circulating tumor DNA. Obviously, we are going to assess safety profile as a very important endpoint for this patient population. So in terms of the statistics behind the study design and sample size, is that we expect that, number one, the response rate will be evaluated by independent review committee assessment, and target lower bound of 95% confidence should be response rate over 30%, but the assumption is that the overall response rate will be 50% with statistical power of 90%. So the sample size, 66 patients with a baseline radiographic assessment will be evaluated for primary efficacy analysis. And we would like to enroll at least 5 pediatric patients, which will be analyzed separately. Now, let me go to how we envisage the study procedures. So it will not be very different to any other cell therapies. The patient will be initially screened. We expect the screening to be completed less than 28 days. Then we'll draw the blood for cell preparation, and if needed, the bridging chemotherapy will be allowed. There will be washout period prior to infusion of the cell therapy. There will be baseline assessment. Prior to a cell therapy, the patient has an active disease, and then the first evaluation efficacy will be based on the Lugano 2014 or Bruce Chesney criteria, as we used to call it. Will be evaluated 6 weeks post cell therapy, and then repeat for a subsequent 24 months. So first question really for pediatric ODAC will be really what is the experience with bendamustine fludarabine. So even in the adult patient population, bendamustine fludarabine is not the most commonly used lymphocyte depletion therapy, although there is a significant experience with bendamustine for Hodgkin, primarily non-Hodgkin lymphoma. So, but the limited number of the pediatric patients who receive bendamustine, they tolerated treatment very well. There's a limited experience in the patient with Hodgkin lymphoma, acute lymphoblastic leukemia, acute myelogenous leukemia, with a safety profile very similar to adult patients. Treatment of the relapse or refractory Hodgkin lymphoma usually use, again, small number of the patients, 120 milligram per meter square for 2 days, every 28-day cycle. In our protocol, we are using 70 milligram bendamustine for 3 subsequent days. But in terms of the PK profile in the pediatric patient population, it looks similar to adults. And fludarabine, there is plenty of experience in the pediatric patient population, is very well tolerated. So now I would like to address the monitoring, primarily focusing on the pediatric patient population. So patients will be admitted for a CD30 CAR T-cell therapy administration as recommended by many panels, published the data in Annals of Oncology 2017, published by MD Anderson in the National Nature Review, Clinical Oncology, etc. Patients will be monitored for 24 hours at least prior to be discharged. When they are discharged, they'll be coming daily to a treatment facility for 10 days, excluding weekends. Patient may remain actually hospitalized, and it will be according to a local practice and to be monitored. And after 10 days, the discharge from the hospital, patient will have to be in a 30 minutes driving distance to treatment hospital that can be monitored frequently. The primary reason for that is highlighted on the subsequent slide. Clearly, there are 2 major concerns. One is the cytokine release syndrome, although we haven't experienced the grade 2 and higher in our program, but that still remains to be seen in a larger patient population. And the cytokine release syndrome in pediatric patient population requires special attention and has to be treated very aggressively. The same goes to central nervous system toxicity. And grading of the CRES according to generally used toxicity criteria for adults is probably not very applicable to infants and definitely not to the young children population. So then we suggest to use our CAPD or CARTAC stand grading system, and that should be performed at least twice a day during the admission. And clearly doctors, nurses, and but maybe the family members, they will have the best experience. The mental status of their children is changing. So pediatric patient population will require the significant attention to two of these major potential risky toxicities. So potential challenges of the clinical development of CD30 CAR-T in the pediatric patient population. So one is obviously to find sufficient number of the patients and treat them by the protocol, on the protocol. That will be definitely very important. Significant attention has to be done to volume of the blood required. Adult medical oncologists, they are very cavalier, and it's easier to draw the blood from adult patients, but clearly the children are not just the small adults, but they have some special requirement, and the amount of the blood which will be drawn has to be very carefully assessed and used very carefully, even more carefully than adults. So what will be very important to leukapheresis for T-cell production, we expect to get 30 ml, and based on our experience, they should provide sufficient amount of the T-cells if we use the criteria to have more than 100 lymphocytes or more than 500 T-cells in the blood. So that will be very important. And what will be important not to only get the leukapheresis that using for infection disease testing and for HLA typing, just to be sure that we can identify correctly the patients. Another aspect what has to be carefully addressed are potential toxicities related to leukapheresis. Children will very likely require the central venous catheter. They might be hospitalized for this. And there are some toxicities associated primarily with citrate toxicity, and has to be carefully calculated by the formulas. The central venous catheter may require the sedation, and then it might be need to provide blood transfusion to patients. Last but not least is in cell therapy, probably more than in any aspect of the cancer therapeutics, the quality and well-controlled manufacturing process is essential, and it's much more complicated than if it's a small molecule or even with antibody. So Tessa is working very closely with the Baylor College of Medicine and UNC, and we are working on to really transfer the academic process, which is obviously done in a GMP spirit, GMP facility, but still required some additional tuning up by industry. So we have to enhance the control. We have to establish a robust quality control and product characterization, quality of the GMP manufacturing facility, and establish a two-tier cell banking to really be sure that we have a high quality and consistent manufacturing process. So with all of this, I would like to complete it saying that it seems to be that in spite of the great success of the Hodgkin lymphoma, and it's really amazing where the field got from 1970s when MAP was published and ABVD today, but still there's an unmet need patient population. The CD30 is a well-validated target for classical Hodgkin and for some non-Hodgkin lymphoma. There are very encouraging data from these two parallel phase 1, phase 2 trials. What is encouraging that the efficacy is very encouraging and safety profile is very well tolerated. The CD30 CAR-T received the ARMA designation. We are working very closely with FDA to really proceed to a pivotal trial. And with all this, I would like to close and thank you very much for the opportunity and for your input.
Thank you very much for your presentation. We will now take clarifying questions for Tessa Therapeutics. Please use the raise hand icon to indicate that you have questions. Please remember to put your hand down after you have asked your question. Please remember to state your name for the record before you speak. It would also be helpful to acknowledge the end of your question with a thank you, and end of your follow-up question with that is all of my questions, so we can move on to the next panel member. So we have Dr. Ballard.
Hi, it's Catherine Bollard here from Children's National, Washington, D.C. I'd firstly like to congratulate Tessa Therapeutics on an outstanding presentation. I do have some clarifying questions, if that's okay. The first one is, I note that there is still a mirroring portion of the single chain. Do you have any data on immunogenicity of that mirroring portion, and if you have any experience with retreatment in the event of that? Secondly, I note that for your pivotal trial, you've decided to select 72 hours as your timing for your baseline scans before lymphodepletion, which is laudable. I just would like to know from your previous data on the other 3 trials if that was always achievable, that 72-hour window prior to getting the scans prior to the lymphodepletion. And then thirdly, do you have any hypothesis why CD30 CAR T-cell persistence was not correlating with response?
Thank you very much, Professor Bollard. I'm really glad these are excellent, insightful questions. So going straight to your question about the HAMA, the limited data we have, there was no HAMA in this patient population. I'm not totally surprised, you know, as was all panel members, that these are, number one, Hodgkin lymphoma is a pretty immunocompromised environment by itself, but over the 7 or many other cycles of the chemotherapy, I'm pretty sure that their immune system is not totally competent. And so, no, there's a limited number. We are going to test it much more carefully and vigorously in pivotal trial, but right now I don't have any information that there is a positivity in terms of HAMA. As the data available to Tessa. In terms of the PET, this is a fantastic question, and you know better than I do that the logistics of arrange it 72 hours prior to a treatment may not be a trivial in many institutions, and we will discuss with investigators more in depth. We probably will have to open as a window 3 plus maybe 1 or 2 days, but it's a very, very good question. In the term of the CD30, the mechanism and the safety profile, I think that why there is no correlation between the persistence and the response rate, it's very hard for me to address, but my speculation is that probably in hematologic malignancies, more than in solid tumors, that probably the peak and expansion of the cells will be more important than persistence. But this is just purely hypothesis. I don't have any data to support it. Thank you very much for the questions.
Thanks. Dr. Pepper, can I ask a follow-up to my question?
Yes, of course.
So for the immune response, that's good about the HAMA. I guess I was more asking about cell-mediated immune response. I'm sure you're aware that there have been HLA-restricted T-cell responses identified to the murine portion of the CD19 CARs that are not humanized. So I was really asking about that. Has that been looked at?
I'm sorry, I missed that. So thank you very much for your clarifying question. I don't have the data to support one way or another. But it's excellent, and we'll definitely look into it. Thanks a lot.
Okay, thank you very much. Thanks, Alberto.
Thank you. Next person is Dr. Kamani.
Hi, thanks, Dr. Papo. This is Nanesh Kamani from Children's National in Washington, D.C. I have a couple of questions, clarifying questions. First, obviously intrigued by the low incidence of cytokine release syndrome likely related to tumor burden. Did you see a correlation between the type of lymphodepletion chemotherapy used and the incidence of CRS? And do you have data on the kinetics of lymphocyte recovery after receipt of the CD30 CAR T cells in terms of how fast the lymphocyte counts recovered? Because that would obviously determine the susceptibility to opportunistic infections post-treatment. And a couple of other questions. you decided to assess the response rate at 9 months. I'm not sure I understood why you chose that as your primary endpoint. And finally, do you have any preclinical data from cell lines or otherwise on CAR T cells for neuroblastoma cell lines? Because I know that a number of neuroblastomas will express CD30. Thank you.
Thank you very much, Dr. Kamani. That's excellent questions. So I will start with the last one. So no, we don't have any data on neuroblastoma. We are really focusing on Hodgkin and non-Hodgkin lymphoma. So, but it's a very intriguing questions, and we should definitely look into it. In the term of the lymphocyte depletion, no, there is no really difference, at least in cytokine release syndrome. Where is a clear difference is the type of the lymphocyte depletion and the skin toxicity. There was almost none in bendamustine, and there was 40% in a flu-cy regimen. So that was clearly a difference. In the term of the lymphocyte recovery, was pretty good, but I cannot give you a timing from the top of my head. But I can say there were no opportunistic infections, so I assume that it was not really. The longest recovery was actually on bendamustine, fludarabine, and it was a 3-month recovery for thrombocytopenia. It was the longest durable, discernible side effect of LD therapy.
Thank you. I have one question, if I may, Dr. Papo. One last question.
Of course.
So I assume you are going to, for the proposed trial, you will have a central manufacturing facility. Is that correct, with the cells being collected at individual institutions and then being shipped to the central manufacturing facility for CAR T manufacturing? And if so, do you plan to have any specific guidelines or quality control for the leukapheresis procedures at individual institutions, and how you intend to make sure that that is generally standardized? Thank you.
So fantastic. I'm sorry, I forgot to answer your question about the response rate. My apology for that. So actually, a response rate, the FDA was asking that we have to have durable responses. So to have let's say 6 weeks as the primary endpoint, when you use it based on Lugano criteria, the earliest sign of the antitumor activity, they would definitely not suffice. So there was a discussion between FDA and Tessa Therapeutics, and we landed on 9 months. So the discussion was between 9 and 12 months. So it's not that the responses is done first time at 6 weeks post-therapy, but then the responses has to be durable. So landmark analysis of response rate will be at 9 months. So that will be the primary endpoint. I'm sorry, I just missed your question. I forgot to answer. In the term of your question about the manufacturing, you are absolutely correct. It will be central manufacturing in the GMP facility. And actually, Tessa is now working with a CRO to audit and inspect all the leukapheresis facilities to be sure that we are using a standardized process. And actually, this will be to some extent addressed where we have, unfortunately, probably mostly virtual meeting with all the sites, investigators and study coordinators will be discussing again. So we will have a pretty strict control in the terms of the quality of the leukapheresis, and actually even the time which we allow from a time of leukapheresis to bring it to a central manufacturing facility, expand the cells, then cryopreserve, return back. That's probably something what will be very important. Primarily will be multi-site, a global pivotal trial study, and we expect to open the sites not only in North America but in Europe as well. So it's very important. Thanks for the question.
Thank you.
Next is Steve.
Steve Du Bois from Dana-Farber Boston Children's. I have a few questions, please. The first is with brentuximab, another CD30 targeting agent, there have been reports of serious pulmonary toxicity, and I wonder if I'm not a lymphoma expert, so I'm wondering, is that thought to be due to the payload of that antibody drug conjugate, or is that potentially an on-target effect? that might be worth monitoring as part of your trial. My second question is if you have encountered cases with progressive disease after response where CD30 has been lost on follow-on biopsy. And then the third question is, what is your recruitment strategy as it pertains to the patients age 12 to 17 years of age? And how is that sort of strategy playing out in terms of the types of clinical trial centers that you are planning to include as part of the trial?
Thank you very much, Professor Dubois. Excellent questions. So let me start with your pulmonary toxicity. I'm not aware that it happened with the naked antibody. I'm not aware that it happened with CD30 CAR. I would be assuming there is more payload related, but definitely will be monitoring patients from all the safety perspectives. In the term of the CD30 loss, that's a fantastic question. I'm sure it's driven by experience with CD19 and CD22. So number one is that 90+% of our patients have failed after the BV therapy, and we had over 60 to 75% complete remission and 88% response rate. It's sort of a clinical justification, must be CD30 there. But there is actually better example. There are post-relapse few biopsies for patients or after BV or actually after CD30 CAR. And in all, the CD30 was there. And actually, I'm not aware of that somebody will do the mutation load. If this patient has a mutation on binding side of CD30, but it seems to be there is not. So with the data available to me, I can say that CD30 loss is probably not a mechanism of escape resistance to a CD30 CAR T, so it's not similar to CD19 or CD22. And in terms of the pediatric strategy, so I'm a big fan of pediatric patients, working on OncoSpar in my previous life. So actually there are several colleagues in various pediatric institutions. So we go after the major pediatric institutions where I hope that we will enroll at least 5 pediatric patients for this study. So yes, we are going very actively, not that going to the typical adult cancer institutions with lymphoma, but specifically to institutions where they have a patient load that they hopefully will be able to support our clinical trial. So thank you very much for the question.
Yes, thank you. Alberto, no further questions from me. Thanks.
Thank you. Ted, you're next.
Hi, Ted Leitch, I'm U2 Southwestern. Steve asked the majority of my questions. I guess the last question I would have was around your pediatric dosing. It looked like the potential the target dose was lower for children if you do 2 × 10⁶ cells versus 200 million cells for adult patients, and I just was curious about the rationale for that.
It's an excellent question. I think we talked to some pediatric oncologists, so we plan to use 5 million per kilo. So yeah, it might be lower dose, but actually did work very well in this in the 2 out of the 3 patients. It's a very good question based on the PK profile. Probably okay, but we didn't in a very formal way study the dosing of the CD30. So we are using the dose more coming from a CD19 CAR rather than our own PK/PD study, which will identify also following more the approved to CD19 CAR. I don't know if I answered the question.
Yes, thank you. No further questions.
Thank you very much. Thanks.
I have a couple of questions. What is the approximate time from the time of leukapheresis to sending the product to getting it back? Just for the panel members to have an idea of how long you would have to give some kind of bridging therapy. The other question I had is, was there any correlation with any of the side effects and response? Specifically, I was very intrigued about what you saw in the skin, that patients that have grade 3 skin reactions had a better chance of responding or not. And then to follow up a little bit on what Steve said, are you investigating any mechanisms of resistance other than CD30 downregulation to try to explain why patients did not benefit long-term, a significant proportion of them, from the CAR T-cell therapy? And finally, are you exploring other enhancements to your CAR T-cell, like adding additional co-stimulatory molecules to improve the activation, survival, and expansion of modified T-cells? Those were my questions.
Well, thank you very much. These are all fantastic questions. So let me maybe start with the vein-to-vein time. So vein-to-vein time is really driven by the two factors, unfortunately. One is to get enough cells, and one to really get the testing. And that's really sort of a testing piece to be sure that you have a quality attribute and you don't have any mycoplasma, any microbiome, etc. That's really something what is under discussion. It depends on what type of the testing they'll be allowed by FDA. That may have the implications of the vein-to-vein time. I would say it's probably around 5 to 6 weeks with all the testing. Can it be shorter? Maybe. That will be definitely part of our discussion with FDA. Now, in the term of the mechanism of resistance, yes, it's a fantastic question for cell therapy, fantastic question for antibody, for immunotherapy, etc. So yes, we will be definitely studying. We have interest in the biomarkers, although it's not really easy to come with the biomarkers for fixed-line therapy for Hodgkin's lymphoma. So that will be something what we'll have to do and look into it. We'll definitely we'll ask investigators to get a tumor biopsy. You, as a treating physician, you know better than I do that it's not easy when the patient has a progressive disease to get a biopsy from every patient. It will be extremely valuable, and we'll be very much interested to really do some deep dives. We have a pretty active biomarker program in our company, so definitely very much interested. Your last question is really a very, very fundamental question, which we are very much interested how to enhance the strength and the quality of the T cells to be even more robust. Yes, we are working on it. Hopefully, in a couple of years, we'll come again to Pediatric ODAC, maybe sooner, to present some of the strategies, not necessarily only with autologous, but maybe with other programs. But at this moment, I think for this particular program, I think we'll stick to CD30 CAR T. We are not going to change the signaling molecule, and we are not going to do any other alteration because that will definitely derail the focus to really go to high unmet medical need patient population as soon as possible. But we are working on it as a very important future programs. Thank you very much for the question.
And there was just a correlation between side effects and response. Did you see any?
I'm sorry. My apology. Actually not. The side effect, so number one is that in terms of the cytokine release syndrome, the majority were grade 1, and majority of responses were complete remission. So I don't think it's really tweaking a very small number. In terms of the skin toxicity, would not be a very good correlate because a significant number of the patients had a complete remission at UNC, and there were no skin toxicity or almost none. So the majority of skin toxicity were driven in cytoxin, fludarabine. So very difficult to really come up with a correlation. It's not like you have a grade 3 cytokine release syndrome, maybe you have a high probability of response. That kind of correlation we didn't see in the relatively small number of the patients. But we'll be monitoring it carefully in our pivotal trial and see what we can learn from it. Thank you very much for the question.
Thank you. No further questions. Malcolm, you're next.
Thank you, Alberto. Malcolm Smith, NCI. And first, thank you for your excellent presentation. My question is whether the CD30 CAR T is being envisioned as providing a path to subsequent auto or allo transplant, or alternatively as a curative strategy for the pediatric patients who might enroll in this study?
So, Dr. Smith, you are reading my mind. Thank you very much for the question. So one of the major interests or big interest in the company is how we can bring this hopefully efficacious and safe therapy to early line of the therapy, primarily for a patient who may have a long-term sequelae like pediatric patients. So yes, the goal is that after we enroll certain number of the patients to look at the efficacy and safety profile and have a further discussion with FDA how this can be introduced to early line of therapy and how the study design will look like. Yes, definitely in our mind. Yes, the second question is true as well. The CD30 CAR is a springboard for our thinking to go to our program. Yes, and actually the study will be hopefully open very soon. So thank you very much for your questions.
Thank you.
Next is Julia Glade-Bender.
Thank you very much, Julia. I apologize if my questions are naive.
I have a difficulty hearing you. I'm sorry.
But,
we're having a little bit of difficulty hearing you.
Can you hear me better now?
Much better.
Okay. So I am not a cellular therapy specialist, but just a few questions. Thinking along the lines of Dr. Smith's question, I know you said that persistence was not predictive of response, but how about duration of response? Because ultimately that is an issue when used in the relapse strategy without a consolidation plan.
Thank you very much, Professor Bender. That's an excellent question. As I mentioned, the study was still censored what we presented, and even the data which were accepted to a Journal of Clinical Oncology, there's still a longer term of follow-up. We'll definitely look at a correlation between the durability of the response and the persistence of the cells. It's an excellent question. Thank you very much.
And one other question, is there a minimal dose at which to expect a response? I did not respond.
Unfortunately,
you're fading again, Julia.
Okay, I'm sorry. Yesterday, I apologize. In terms of minimal dose,
it's still very choppy. Julia, are you still on the line? Okay, we will come back to Dr. Glade-Bender in a minute. Next is Nita Seibel. Nita? Okay, next is Dr. Riemann.
Thank you. Thanks, Alberto. Dr. Horak, thank you for an excellent presentation. Just a couple of questions. Can you clarify that your pivotal study will not proceed until the commercial manufacturing process has been established? And if that is in fact the case, do you have a timeline for when that should occur?
Thanks a lot, Professor Hernandez. Excellent question. Yes, we plan to do with a commercial manufacturing process. We are working with, unfortunately, COVID-19 changed our life in every aspect, not only in the study conduct, but in the preparation, manufacturing, etc. So we are targeting the end of the year, but it really, many things can change between now and then, and depends how the COVID-19 will proceed in Europe and in the United States. So the target is to open it towards the end of the year, and I hope that we can stick to it. We are all very enthusiastic and working very hard to stick to the timeline.
Okay, thank you. Another question relates to the number of adolescent patients that you plan to enroll on this trial. Why was the number 5 selected? Number 1. Number 2, do you think 5 patients is sufficient for evaluation of efficacy, even though much of the efficacy could be extrapolated from the adult experience? But only 5 patients is a pretty sparse number for even assessment of short-term and long-term toxicity?
This is an excellent question. It's a very thorny question. Unfortunately, I don't have any good justification for number 5. I think the concern, we want to have a pediatric patients in the study. And we have a history to really work very hard to include from as early as possible children in our drug development. A concern which I have specifically is that it's really due to a high curability and relatively low incidence of the Hodgkin's lymphoma under age 12. The number of the patients we can enroll will not be very easy. We are very much open-minded to hear the input from pediatricians, what will be a reasonable number of the patients to be treated to get the access, not only the efficacy, which I agree can be extrapolated from adults, but primarily the safety and long-term safety. So that will be fantastic to get the input from pediatric ODAC. So it was arbitrary pick up the number 5, I have to admit, and the age 12 again was really coming more from discussion with some investigators that the probability to get a younger patient population might be even less likely. But again, we'd love to hear the opinion of pediatric ODAC.
I think it's a somewhat disappointing arbitrary number. I would encourage you to think bigger. And I guess I would also question the age of 12, although, as you pointed out, the majority of patients with this disease are adolescents. But that doesn't mean that 100% of children less than 12 with classical Hodgkin lymphoma are cured. The other factor to consider here is that the real unmet need, I think, in classical Hodgkin lymphoma is not just in salvage therapy, but in frontline therapy that may, in fact, be more efficacious, less toxic from the standpoint of cardiac toxicity and second malignancies related to radiation therapy, which are significant factors in the management of Hodgkin lymphoma. So I would just encourage you to think a little bit more extensively about the pediatric population as far as numbers and as far as the age groups that you would want to assess these cell products.
Thank you very much, Dr. Raymond. Excellent point. So I have to admit that we are all thinking how to bring the CD30 CAR to early line of therapy, primarily for pediatric patient population, for elderly patients who do not tolerate high-dose chemotherapy. Unfortunately, I do know that the long-term sequel of the chemotherapy, primarily high-dose chemotherapy for children, might be devastating over the lifespan they hopefully have ahead of them. So the question will be really, what is the minimum number of the patients one would need from a late line of the therapy to be courageous and to go to second line pediatric patient population? And that will be really very interesting discussion. Definitely companies very much interested to think big and think how to push CD30 to second line primarily in the children. Yes, I 100% agree.
Okay, thank you.
Thanks a lot for the question.
I'll try to go back to Julia. Julia, are you on the line?
Yes, I am, but I don't know if you can hear me any better.
Much better. So go ahead with your question.
Okay, thank you. I was just going to ask about a minimal dose, effective dose, because I did notice that the one child with progressive disease had received a log fewer cells.
So this is an excellent question. Unfortunately, it was not robustly studied, I would say, even for adult patient population, definitely not for children. So yet the general perception is looking at the peak of the cell expansion, and the persistent is cell dose dependent. So I would assume that the more is better in this case, primarily when the treatment is very safe and very well tolerated. But what is the lowest level? I really cannot answer the question. The lowest study, to my knowledge, and maybe Professor Bollard knows this better, I think it was around 20 million. And that was borderline efficacious. So I think that when it comes to a dose in adults, around 100 million, the efficacy is going up. Is there a cap in a term of the response? I really don't know. The Tessa plans to open this year the study in a non-Hodgkin lymphoma, initially as a dose escalation study, in a very formal way. We'll start the dose which we are using right now for Hodgkin, 200 million cells per meter square, and try to dose escalate and see if there is any dose dependency. But based on the data available to us, unfortunately, I'm sorry, I cannot answer the question, what is the lowest level? But probably we don't have to worry too much about the lowest level as long as we can get a T cells, which generally we were very successful, or our academic colleagues are very successful to generate sufficient number of the T cells. And since the safety is good, so probably don't have to go lower. But again, I don't think that in children it was in a formal way studied, and adults, very limited data in 2 or 3 patients per cohort. I'm sorry.
Thank you very much. Thank you very much. I was just afraid of underdosing children. That was all. Thank you so much.
And if I may give sort of addition, that's the reason why we are doing a very formal dose escalation study in non-Hodgkin lymphoma. Worried that the patient with a larger tumor volume or tumor burden, they may benefit with a high dose. So again, to address your question, to prevent the underdosing patients. But thanks a lot for your question.
Okay, we have Catherine Bullard.
Yes, sorry, just to build on some of the other comments, especially about durability of the response. Do you have any data in patients who've been retreated with the CD30 CAR?
It's a fantastic question. I believe there are a couple, and actually very, very short-term duration. One patient has a stabilization, and one patient has a complete remission, which was very short duration. I think a few months.
Thank you.
Thank you very much.
And Donna?
Thank you very much for this presentation. I would reiterate what Greg already brought up about considering lowering the age without assuming that all of those under 12, are going to be cured first time around, and also this issue about bringing this forward to frontline to reduce toxicity. I had a question. I was intrigued to see that you had an allogeneic CD30 CAR that's in development using EBV-specific T cells, and I was curious about your development plan, how you see that fitting into this particular landscape.
It's a fantastic question. One of my favorite. Thank you very much. Going back to your first question or first comment, I think that really the goal, when we talk about the 5, and I understand Dr. Raymond, his concern about the small number of the patients. We just would like to see the safety is really retained to be as good as we hope it is, and open it to age pretty much as low as it can go. So I don't think that after we enroll a few patients and we feel more comfortable, we are more than happy to amend the protocol and go to a lower age. So that will be sort of a first answer. I think we are working on the development plan for CD30 allogeneic DSTs. You're right, there is a lot of very exciting hypothesis behind that strategy, which was developed by some people around the desk, around this ODAC and at the Baylor College of Medicine and Sloan Kettering. So clearly a lot of interest in this technology. We plan to first go after CD30 positive lymphoma, and not really specific for Hodgkin or non-Hodgkin. And then based on activity, whatever we observe in the phase 1 program, we'll make a determination where to go with it, in which subset of non-Hodgkin lymphoma or Hodgkin or non-Hodgkin lymphoma. I think that it's very likely that our CD30 CAR will be significantly ahead of allo program, and therefore it's very likely that that will be the first to, if we decide to, and data support it, to push it to early line of therapy, which trust me and trust the company. We have a significant desire to bring it earlier. I mean, unfortunately, I witness long-term toxicity from Hodgkin lymphoma chemotherapy, and it's definitely not pleasant. So I fully agree. It's an excellent question and suggestion. Thank you very much for the questions.
Good, thank you. No more questions.
Okay, Nita, I see your hand still up. Would you like to ask a question?
Yes, I have two questions. Nita Seibel from the NCI. First of all, have you seen any development of the CAR-HLA syndrome that's been described after some of the other CAR-Ts, such as CD22, that follows the cytokine release syndrome? And then secondly, can you expand or give more description about any cardiac toxicities that you've seen?
So the second one is easy. I cannot describe any cardiac toxicity. But can you clarify? I'm not really sure I fully comprehend your first question.
Well, there's been a description, particularly after CD22 CAR T cell, that after cytokine release syndrome, that they've seen what is being sort of labeled as CAR-HLH, or hemophagocytic lymphohistiocytosis.
None.
None? Okay.
No.
Okay.
None. It's a very, very good question. Thank you very much. I'm sorry. I just didn't comprehend what you're asking.
Sure.
Sorry. Thanks a lot.
Thank you. Thank you.
And I think we have one last question. Greg? Greg, do you have another question?
No, I'm sorry. I didn't put my hand down. I apologize.
Okay, so if there are no additional questions, we will move to the next portion of the meeting. Today there is no open public hearing session, so we will go straight to the charge and questions to the subcommittee and panel discussions. After each question is read, we will pause for any questions or comments concerning its wording. Then we will open the question for discussion. So I will ask the FDA to read the first question, please.
Hi, this is Megan Zimmerman with FDA. The first point for discussion is pediatric age groups include neonates from birth to age less than 1 month, infants ages 1 month to less than 2 years, children ages 2 years to less than 12 years, and adolescents ages 12 years to less than 17 years. Please discuss which pediatric age groups are candidates for study with CD30 CAR-T and which can reasonably be excluded.
Okay, so if you want to raise your hand and try to address these issues. Let me see. Malcolm, you're first.
Thank you, Alberto. Malcolm Smith, NCI. Regarding the age range that might be studied with the CD30 CAR-T, I think it's important to take a reality check on how many patients might be able to enroll. To a first approximation, the number of patients who might be eligible for the study each year would not be much higher than the number of patients who die from Hodgkin lymphoma each year within the age range, since this is a treatment that's given after failure of the likely curative therapeutic options. When you look at mortality data for the U.S. for the most recent 5-year period for which we have data, and for patients who are less, or children who are less than 15 years of age and deaths attributed to Hodgkin lymphoma, it was 2 per year. Looking at 15 to 19 years, the deaths attributed to Hodgkin lymphoma was around 7 per year. And so I think it's going to be really challenging to enroll, certainly less than 15. And so I think you can pick a number, 5. It may be desirable to get more than 5, but 5 may actually be a realistic number. The second comment I would make in terms of moving this to frontline and because that was discussed during the presentation and moving into frontline in the pediatric setting, I think here as well a kind of reality check. The activity of checkpoint inhibitors in Hodgkin lymphoma has generated this whole new line of promising therapeutic research, and there will be a generation of clinical trials attempting to incorporate these agents into the frontline setting. And these trials, I'm sure, will make attempts at minimizing or avoiding radiation exposure for as many patients as they can, as well as ameliorating other long-term effects. And so I think, a few years from now, we'll need to check on the results from these studies and the anticipated long-term effects based on the chemotherapy and radiation doses using these studies to think about how, what are the needs for this patient population. Thank you.
Thank you, Malcolm. Catherine?
I thank you very much. I agree. It is Catherine Bollard here. I agree with everything that Malcolm just said. The other caveat I would just add to the children age group, or even less than that, is there is a percent of children in that group who present with Hodgkin's but actually have an underlying immune deficiency that needs workup and probably allo transplant. So just to be aware of that as well.
Thank you. Julia?
Just to advocate for access, while I don't think it should be required, perhaps it could be allowed for children ages 2 to less than 12. Again, I'm thinking actually more of anaplastic large cell lymphoma than of Hodgkin's lymphoma. In my recent experience, I've had several younger children that, despite the novel therapies, still have a very difficult time with the anaplastic large cell lymphoma. So perhaps widening eligibility for younger children, at least to get experience with this drug, might be of interest.
I think that's going to be one of the questions that is coming, so we can address that issue when that question comes up, where we can incorporate this into other malignancies that have CD30 abnormalities. And then Ted.
Hi, Ted Leitucci, Southwestern. I was just going to say something similar to what Julia said, which is that I agree completely with Malcolm that the number of patients, especially in the lower age ranges, will be very small, but I don't know that there's a strong rationale to put a hard cut at 12 years for eligibility if you're already using weight-based dosing, et cetera, for patients older than that. And so I would hate to exclude, an 11-year-old with Hodgkin's who might potentially benefit from this therapy. Obviously, the infants and neonates are a different group, and I think certainly would not be in the Hodgkin's cohort.
Thank you. I don't see any other questions. So if there are no additional questions, I will try to summarize the discussion for question number 1. So it appears that the mortality rate for Hodgkin's is very low, especially in patients that are less than 15 years of age, and therefore there may be a challenge into recruiting into this clinical trial, whether it's 15 years of age or older, or 12 years of age or older, or if it's going to be age weight-based. If a patient is, for example, 11 years of age but meets the weight criteria, they should be allowed to be enrolled on the clinical trial. Regarding the use of this therapy as frontline, I believe that we need to await the results of other clinical trials that are ongoing that may show promise with incorporation of immune check inhibitors in patients with Hodgkin's disease. And finally, we will discuss a little bit more in one of the next questions whether additional histology should be included in this kind of trial and whether the age range should be modified based on the histology. Please let me know if I left anything out or if it's okay to move to the next question. Did I summarize everybody's comments accurately? Sounds like a plan. We're going to go to question number 2.
This is Megan Zimmerman with SEA again. The second question has 2 components. Please discuss the variability of the preparatory lymphodepletion therapies and their potential applicability to the pediatric population. And please discuss CD30 positive malignancies other than classical Hodgkin lymphoma, which could be studied in pediatric patients.
Okay, Julia, you go first.
I apologize, I had not put down my hand, but here is where my point is relevant. I think we need experience in patients with other diseases like anaplastic large cell lymphoma, and that might involve children that are younger in age than those with recurrent refractory Hodgkin's disease.
Thank you. Malcolm?
I'm sorry, Alberto, I put my hand down from the previous question.
Okay, I'm sorry. Ted?
Yes, thanks. This is Catherine Bollard. Firstly, discussing CD30 positive malignancies other than classical Hodgkin's. Certainly, pediatric ALCL is CD30 positive. COG has currently completed a study with upfront use of brentuximab in this setting. And the problem here is that most pediatric ALCLs also express ALK, so the ALK inhibitors such as crizotinib have high efficacy here, even in the single agent setting for relapse disease. So while Dr. Smith explained the small numbers of relapse refractory Hodgkin lymphoma patients in the pediatric setting who would be eligible, I would argue that it would be even less in the pediatric ALCL setting, which would make this challenging. I do think there is some concern about the variability of the preparative regimen. Bendamustine and fludarabine, this is the only setting that I'm aware of using CAR T cells where you've used that, where this has been used as a lymphodepleting regimen with bendamustine, fludarabine. To my knowledge, there's no data using fludarabine bendamustine alone in combination with children, or even I'm not sure about adults either. So this would be data that may be important. That being said, the safety profile of your approach seems extremely reasonable, and given the data that you have so far, it would suggest that you should go with the standard lymphodepletion regimen of bendamustine, fludarabine, in my opinion.
Thank you very much. Ted?
Apologies, Alberto, I put my hand down.
Thank you. Dr. Kamani?
Yeah, thank you. This is Nanesh Kamani. So in terms of the preparatory lymphodepletion therapy, I agree with Dr. Bollard that I'm not aware of bendamustine having been used in children as a lymphodepletion regimen. And this would be probably the first setting where it would be used. Fludarabine, obviously, we have a lot of experience with it. But I agree with her that based on the prior experience with bendamustine and fludarabine, that would be the regimen that seems the one that we should go with for this trial. In terms of the other malignancies, since we're talking about a very limited number of patients that are proposed, I doubt that there will be sufficient information to reduce effectiveness in Hodgkin's lymphoma per se. Based on that, I would consider allowing younger patients, definitely those between 2 and 12 years of age, with other indications such as ALCL, to be entered only to allow us to gain more experience with that age group as well. In terms of the ability to collect cells, that should not be a problem per se. So I think because the current indication is classical Hodgkin's lymphoma, but the number of patients that are being suggested is not going to tell us a whole lot about efficacy per se. I think I would include younger patients if they qualify based on their diagnosis, whether it's ALCL or other indications. Thank you.
Thank you very much. I do not see any other hands. Does anybody? Dr. Cheng? I'm sorry, Dr. Cheng.
Hi. Yes, hi, John Chang, industry rep, and I'm with Merck. I had a comment and then maybe a question for the panel. Oftentimes in a study as being proposed by Tessa, it's an adult Hodgkin's lymphoma study, and along adolescence, I think is a desirable thing. However, when you start to go into pediatric patients with lymphoma, it causes some operational challenges because oftentimes those are unique cooperative group, COG sites or children's sites. And so choosing the site gets to be a little bit trickier once you get to a smaller age, to the younger pediatric population outside of the adolescents. And so oftentimes you're limited in the choices of how many sites you can go and your footprint that you want to use. And so I'm interested in the panel's perspective. How can, or do they have any thoughts as to how to help industry be able to enroll not just adolescents, but younger patient population, knowing that oftentimes it's different investigators, different sites, and different clinics. And so oftentimes industry will choose a adolescent cutoff rather than a pediatric cutoff for that reason, not because we don't want to understand it in younger patients, it's just that it's operationally challenging to have those two pieces in the same study.
Does anybody want to tackle that question? Steve? Go ahead, Steve.
Yeah, this is something that's extremely important. So I'm glad you're bringing it up. Sort of, it's kind of meaningless for a trial to be open, like to down to age 16, 15, 12, or younger, if it's exclusively, if the sites that are participating are exclusively large medical oncology centers. So I think what I encourage sponsors to think about is centers that have kind of both a robust pediatric program and a robust medical oncology program under the same roof. So, one IRB, one contract, and investigators who can participate both on the pediatric side and on the medical oncology side. But it's very, it's really a key question and a really tricky one.
Julia, do you want to add anything to this?
Yes, Julia Glade-Bender, Memorial Sloan Kettering. I want to second what Steve said, but I think what I am advocating here and what I think that Dr. Nanesh is advocating here is really, Dr. Kamani, I'm sorry, that is it possible, rather than to require the accrual of a certain number of patients within an age group, just to have it open but not have it hold up the whole study if it doesn't accrue, just to maintain access so that we could learn more about safety in the